In this video, I'd like to discuss the difference between the blood flow through the brain and through the liver, particularly about the blood-brain barrier so many people are curious about. And I'm bringing the liver into the story because basically the liver and the brain live on opposite sides of a continuum of capillary structure. So capillaries are the teensy tiny little part of your blood flow system, of your heart tree, which starts out with big old arteries and big old veins and ends up at the periphery with teensy tiny little pathways that we call capillaries. And I've drawn two different types of capillaries here on the board. Now, red blood cells flow literally one by one through a capillary. That's how small they are, because red blood cells are really tiny. So in the liver, we have what are called fenestrated or sinusoidal fenestrations, big fancy word for got holes in it. So a fenestra is like Italian or Latin for a window. So the capillaries of the liver have windows in them. Now, why is that? Because in places like the liver and, say, the spleen as well, we have these sinusoidal fenestrations because those organs are basically filters of the blood. So as a little red blood cell goes tumbling along through a liver capillary, it literally falls out of it like water through a colander and splashes about on the tissues of the liver, which is like a sponge soaked with blood, whose chemical intelligence processes that blood and other substances in the blood in a way that ultimately, say, breaks down toxins and protects us from them, or sequesters those toxins into the bile and excretes them. So very handy that the capillaries of the liver are fenestrated, meaning having windows through which relatively large structures like a blood cell can pass through the epithelial lining and through veritable holes in the wall for them to be processed. Now, the brain is the total opposite of that. The blood-brain barrier is basically describing the structure of the capillaries of the brain. So the structures of the capillaries of the brain are featuring very tight junctions in the epithelial lining cells, or the endothelial lining cells of the capillaries of the brain. So look, there's no way out of this thing. So how does anything get through? Well, the red blood cells don't get through. Much smaller things can pass through, gases and ions and such. And if something larger, some kind of protein has to get through or whatever, it's mediated by transporter proteins that are like bouncers at the bar. So this bar is like anybody can come in, it's some kind of speakeasy, and there's no limits to what can pass into the bar of the liver. But the bar of the brain has got bouncers standing around there like, you're not going in there, you're not going in there. Well, you can come in, but we're going to hook arms and I'm going to literally walk you into the bar. So that's how you get across these tight junctions. Or there's also, chemical events that can be communicated through the blood that actually will, for certain substances, sort of loosen the tightness of the junctions and allow things to squeeze through. So it's very specific. Oddly, alcohol gets a pass. So this is the funny thing about drugs and alcohol. Some of them, their effect is possible because of the ease with which they pass through these tight junctions despite the brain's best efforts. So we can have the passage of certain things through here that affect the brain in all kinds of crazy ways because they can transport themselves across the blood-brain barrier. It's like they go to the bouncer, hey, look that way, and they're like, and they go right in. So blood-brain barrier is generally a very effective protector of the tissues of the brain from all sorts of substances that might harm it, whereas the sinusoidal fenestrations of the liver capillaries are basically designed to handle those toxins specifically and break them down. I think that's cool. There are also structures in between these, so it's not all exactly like that. You could have, say, fenestrations that have kind of a proteinaceous stringiness across them that makes it a little harder for stuff to come out, but allows larger molecules to pass through for filtration in, say, the kidney. So there are, kind of gradations between these two, but I give these two as the extreme examples to help you understand, particularly the blood-brain barrier. Thanks for watching. If you'd like to study more with me, go to gilhedley.com. There's a ton of stuff there. Enjoy.